Another important tool in genetic engineering and molecular biotechnology are the microarrays. When we think of microarrays, think of molecular microscopes. We're able to take a very close look at unique DNA patterns or genes using a very interesting combination of technologies. Microarrays are often called labs on a chip. We can take a very tiny amount of DNA and essentially using DNA probes attached to a solid matrix like plastic or glass, like a microscope slide. We can then take our patient sample and after it's been digested with restriction endonucleases, simply expose the microscope slide to the sample and see where the DNA probe and pieces of the sample have hybridized. We're also going to use some type of fluorescent or chemiluminescent tag of different colors, and based on the types of hybridization that occur, we can often determine both genomics, what kinds of genes that are inherited, but also even proteomics, how the genes are expressed and used. The creation and the development of microarrays has rapidly expanded within the last few years and dramatically changed how different drugs and pharmaceuticals are administered, what kinds of combinations. We even have a better understanding of drug interactions with the use of microarrays. One of the early applications of microarrays was to take a look at drug metabolism. Now, in the human liver, there's a cytochrome called cytochrome P450 that's integral in the process of drug metabolism. And with microarrays, scientists and pharmacologists have been able to get a better idea of the wide variety of genetic profiles that are out in nature amongst human populations and how people essentially modify or metabolize different kinds of drugs. Because as you might know, not everyone can take the same drug and have the same effect. There are various P450 genotypes. Some have deletions in the nucleotide sequence, some have duplications, and a couple of examples are the CYP2D6 and the CYP2C19 genes that can be found in nucleotides needed for the production of cytochrome P450, which then lead to a cytochrome P450 that have varying degrees of drug metabolism. It's been determined that about 25% of all prescription drugs are metabolized by the cytochrome P450 complex. And of course, variations of cytochrome P450 will make a difference in how the drugs are metabolized. Now, in terms of microarrays, the ampli chip by Affymetrix is probably the best known. There are other kinds, but ideally what we want to try to do is to develop a drug treatment that's more individualized with the proper dosing and the proper medication for every patient. So we have personalized medicine. I'm not certain how far or close we'll get to that particular target, but we do have a better idea of the variations in the P450 gene, and then the proteins that are produced from those P450 genes that make the P450 complex found in liver cells that are responsible for metabolizing different kinds of drugs. Based on the variations of the P450 gene and then the protein complex that's produced from the P450 gene, scientists have identified essentially 3 or 4 different groups of drug metabolizers. There are some that are poor, some that are intermediate, some extensive, or ultra-rapid. Essentially, a poor metabolizer would be someone who would ingest a drug and not metabolize it at all, thus making the drug ineffective. Intermediate would often be those folks that have sort of the ideal or optimum metabolism and use of the drug. And the ultra-rapid metabolizers simply break the drug down too quickly for the drug to do any good. So the drug is properly metabolized, but too quickly released. Based on the type of mutation a person may have inherited for their cytochrome P450 genes, will often determine what category of metabolizer they will be when investigated with a microarray. So ideally, with the microarray, we could more closely match up the proper drug to each individual patient. Now, in addition to looking at DNA fragments, or using the microarray to determine a pattern of various kinds of mutations and slight changes that a person has inherited, we can also look at proteins, different kinds of pieces of tissues. Microarrays now have been applied in a wide variety of different areas to take small samples and look at very discrete, unique characteristics of both nucleic acids, proteins, and even some molecules in between, like carbohydrate binding proteins, or proteins that have unique characteristics, or lipids, or sugars attached. Simply a nice, interesting, and easy, though technically quite sophisticated way of thoroughly investigating the molecular biology of virtually any kind of organism.